this is the thing.
That should give you forward and back, and that's the pointer.
Okay.
And hopefully that will work out okay.
Great. This time has already started.
So it's 3 o'clock, and I think we're going to try and start as promptly as we can. So I'm Jenny Rowntree, I'm chairing this session this afternoon. And without further ado, really, I'm going to introduce you to Nick Isaac, who's going to talk to us about the impact of neonicotinoids. Thank you very much.
Thank you. Thanks for coming, folks. I know there's tough competition this afternoon. So, like most of you guys, I usually like to save my most recent and unpublished work for the BES. But this year, I'm making an exception, and I'm going to talk about some published work. And there's really 3 reasons for that. One is because all my unpublished work is currently being presented by other people, including just in there, which is kind of annoying, and I realize that actually most of the exciting work I do is actually done by other people. The second reason is that this paper that I've just published is on neonicotinoids, which seem to be of great interest at the moment. And the third reason is that it's a rather new experience for me to publish in the tabloids, and therefore I'm going to shout about it as loud as I can. So why do we care about neonicotinoids and bees? Why is it such a big deal? Well, let's start by looking at this field. This is what most large parts of southern England look like nowadays. This is an oilseed rape field, of course. Oilseed rape has become one of the most widespread crops used in England, and particularly in southern England. And the line on this graph here shows that the total area covered by oilseed rape and how it's increased over the decade 2000 to 2010. So there's a massive increase in the use of this crop. But this crop could not be grown in this country without a really large use of pesticides. It's an absolute magnet for all sorts of pests. And so the bars in this figure show the use of a particular class of chemicals called neonicotinoids. And there are 3 or 4 shown here. The main one is imidacloprid, and that's the main one that's going to be contributing data to what you see today. But then more recently, we've got these other 2 compounds, thiamethoxam and what's the other one? Clothianidin. Yeah, I can't read. Good. So what does this mean for us and for bees? Well, these chemicals are applied to the seed, as shown here on the right. They coat the seed, and the pesticide is then expressed systemically throughout the whole of the tissue of the plant. And this is a good thing because it means you don't have to apply large amounts of spray, and so you don't get runoff. And actually, in terms of water quality, it's a really good news story. But it also means that beneficial insects, such as bees, have the potential to be exposed through pollen to the pesticide. So the question is, do these chemicals harm bees? And we're particularly interested in whether they harm wild bees. There have been some studies done on honeybees, but wild bees, relatively little is known about them. This figure illustrates the fact that quite a number of our native wild bees are very important pollinators, or at least flower visitors, of oilseed rape crops. And so I think there's about 20 or 30 species of wild bees here, out of around about 250 of our native wild bee species. So we're interested in wild bees. And there's some good evidence growing now that wild bees have declined over the past few decades. This, for anyone who was at my talk last year, this is the figure I wasn't allowed to show. It's the 2015 indicator of pollinating insects that I put together with some colleagues. And it shows how bees and hoverflies have changed in their distribution over recent decades. So we've got a picture of declining bees, and we've got some concern about these pesticides. And there's some evidence now from a number of studies of negative impacts of neonics on honeybees particularly, and also on 2 species of wild bees, the buff-tailed bumblebee and the red-tailed mining bee, I think. All of that evidence comes from lab studies and a number of medium-sized field trials. We know very, very little about whether these effects scale up to long-term, large-scale population declines. So that was kind of the starting point for our study. So we got some evidence. We wanted to know whether there was evidence of a large-scale, long-term, and large numbers of species. And one of the contexts for this is that because of the evidence for negative impacts on 3 species, the EU imposed a moratorium, and for the last 3 years, farmers have been prevented from using seed-treated neonics, at least without a special license. So it's a very contentious subject. The seed companies and the conservation organizations have very different views. So can we actually fill this with some evidence? So in order to do that, we need to figure out what data we've got. I work in the Biological Records Centre, so I work on biological records. These are data gathered by expert amateur naturalists, such as shown here. These are people who go out into the countryside and they return lists of bee species that they recorded. And they submit these records to the Bees, Wasps and Ants Recording Scheme. And I collated together 86,000 records from a 17-year period, as shown here. It's really noisy data. It's not a systematic survey. People tend to go where they want to go. They tend to survey the species that they want to survey. So we spent a lot of time in my group over the last 2 or 3 years thinking about the statistics of how to analyze those kinds of data. So I'm not going to say anything more other than we have thought about it, and underpinning the main statistical model is a description of how people collect their data. So we've got some data, and we're going to be modeling 62 wild bee species. We have some data on oilseed rape, and we have some data on neonics too. There are 2 key data sets that we were fortunate enough to have access to. One is the June Agriculture Survey. This is an annual survey, and we were able to get the coverage of oilseed rape and reproduce the figure I showed you earlier with a massive growth in the coverage of oilseed rape. And then the second data set is what's called the Pesticide Utilization Survey. And this is a survey that goes out to farmers every 2 years. And from this, we were able to get a measure of what proportion of oilseed rape was treated with seed-treated neonics. And we interpolate that to get a measure that we can compare with our oilseed rape cover, and then we can estimate oilseed rape and neonics for each one. Okay, so what do? We're fitting a statistical model of the persistence of bee populations from 1 year to the next. And what we're interested in is a correlation between persistence and 2 things. The first is how much oilseed rape there is. We expect that more oilseed rape is going to be good because it's a mass flowering crop. It's a huge resource. So we expect populations to benefit from increased cover. And a negative correlation we predict with neonics. And we're going to be comparing 2 different types of species, those that do forage on oilseed rape and those that don't. And we're expecting to find a big difference between the 2. So the first question then is, do species that forage on oilseed rape benefit from increased cover of this crop? That's what we expect to get. And so what I'm going to show you in the figure is that our estimate of the steepness of this line, this correlation coefficient. So we've got positive correlations on the right, negative correlations on the left. So oilseed rape foragers in blue, our best estimate is that they have benefited. The correlation is positive. And the distribution is the distribution of our estimated effect sizes. So non-foragers, their distribution is centered on 0. So there's absolutely no evidence for an effect of oilseed rape cover on species that don't forage on it. So that's good, because that means our categorization makes sense. So we can be really, really confident that species that do forage on oilseed rape have benefited, and those that don't haven't. So then we can move on to the second question. Are neonics harmful? And is foraging on oilseed rape the main mechanism by which species get exposed? So same figure, same sort of figure, positive correlations on the right, negative on the left. And it's slightly more complicated in this case because both groups show a negative relationship. Both have a negative correlation on average. But it's much more negative for the foragers, the blue group. The mean correlation is 3 times more negative. So it does support this idea that foraging is the main mechanism, but it's not the only mechanism, because these non-foragers are also getting a negative hit from increased exposure. So there's this theory that dust caused by sowing is getting elsewhere, and maybe that's why these species are getting exposed. So I've talked about correlation coefficients. What does this actually mean in terms of persistence in the wider landscape? So effectively what I've done here is get the fitted values out of the model. And I've selected 3 species to show you in red what actually happened. This is their long-term trajectory before and after the arrival of neonics in 2002. And the blue is what our model says would have happened had neonics been completely benign. So there was no negative effect at all. So in all 3 cases, the blue line is above the red. So the species, we can infer that it has declined, and that decline is attributable to neonics. The hawthorn mining bee is one where we've got about, we estimate it would have been 18% more widespread without neonics, or a benign neonics. Lazy glossum, 23%. So although it's not a very common species, not very widespread, we estimate quite a big effect. I put this species on the right, the red mason bee, because that's one of the 3 species for which there was lab and field evidence of an effect. And actually we find nothing at all, really. So, what does all of this mean? So, we knew that there were some negative effects, but what we've been able to show is that these do scale up to large-scale population declines over a decade or more. And that that feeding on oilseed rape is clearly implicated as the main exposure mechanism. It's not the only mechanism of exposure, because there's some residual variation we can't explain. For some species, there have been some quite dramatic declines that we can attribute to neonics, and the most extreme case I haven't shown you is up to 30%. We think that this fills a really important gap in the evidence. When the moratorium is reviewed, there will be a lot of different sources of evidence to assess whether these chemicals should be allowed. This is one piece of evidence, and we think it's quite an important one. That's all I've got, so I'm very happy to have your questions.
Great, perfect timing, thank you. Yes.
Hi, so different bee species have different seasonal activity windows, and also rate flowers at a specific time. Well, there's 2 flowering times, aren't there? So did you notice any kind of relationship between species that were foraging around the time of species that were foraging?
That's a great question, and the answer is no, we didn't. We wanted to keep it simple, and so we didn't really look at that. And that would be an obvious reason why some species show... when I plotted those histograms, I was really only showing you the average of all of the species that forage and the average of those that don't forage on oilseed rape. But there's a lot of heterogeneity, so we didn't explore that. But that's an obvious reason.
In your species categorization, about 15 of the species that you use were parasitic bees. Now, the fitness of a parasitic bee is going to depend not only on its exposure to pesticides, but also on the frequency of its host. And you had a group that were of its hosts that were in the group that foraged, but then your parasitic bees were split between ones that foraged and ones that didn't. So I just wanted to know, why did you include parasitic bees in the analysis at all? Because it's an additional confounding factor. And why were they in different groups to their hosts?
That's a great question. I think I would probably have to take that up with you afterwards, because I didn't do that classification. that was something that my co-author did. I'm afraid I don't have the answer to that. The answer to your question of why there's a different categorization, or why there could be, why we include them in the first place. So a species that is a cuckoo can be exposed if its host is foraging and bringing in pollen that is contaminated. So that's the reason why we included them. The categorization, I would have to take that up with my author.
I'm afraid we're going to have to move on. So any other questions you have, if you could ask Nick after the break, please. Thank you very much. So our next speaker is Penelope Whitehorn, and she's going to talk to us about buzz pollination and pesticides.
Hello. Well, I've had a very nice introduction to neonics already. I'm going to be continuing the theme, talking about research we've been doing with bumblebees, looking at buzz pollination and the impact of these chemicals. I've been doing this work with Mario Vallejo-Marin, who's a plant evolutionary biologist at Stirling, and also Claire Wallace, who's an undergraduate student who's been working very hard on the experiment. So to start with some background, many species of plants, approximately 20,000, do not give away their pollen freely from the surface of their anthers. They keep it contained within and only release it through small pores in the tip, so the anther's effectively like a tiny pepper pot. And to get at this pollen, the pollinators need to shake the pepper pot, and they do this with vibrations or buzzes, which is why it's known as buzz pollination. And the ability to buzz pollinate is almost entirely restricted to bees, most notably bumblebees. One species of hoverfly is also known to be able to do it. And interestingly, honeybees are not able to perform this pollination. And plants that need to be pollinated in this way include some very economically important crops, for example, tomatoes pictured here, and also potatoes, aubergines. So buzz pollination is quite a complex behavior. After landing on a flower, the bee needs to curl her ventral side right around the anther, and she grips onto the base with her mandibles. She then needs to generate a vibration without actually beating her wings, and she does this by decoupling her wings from the indirect flight mechanism and then rapidly contracting the thoracic muscles. And this produces a vibration which resonates throughout the anthers, shaking the anther and releasing the pollen onto the bee. And we can find out a lot more about this type of pollination behavior by actually analyzing the acoustic properties of the buzzing sounds. You don't need to worry about the details of that graph at all. It's just a visualization of a 6-second long feeding buzz by a bumblebee in a sound analysis software program. And so these buzzes, the vibrations produced by bees, can be characterized by 3 main properties. Firstly, the duration of the buzz, which is typically 0.1 to a few seconds long in bumblebees. And then there's the frequency, so this is the speed of the vibration, and this determines the pitch of the sound. So in this picture here, the peak frequency is this tallest peak here, and if you read down to the x-axis, it's approximately 330 hertz for this particular buzz. And then finally the amplitude, which is the size of the vibration, and this determines how loud the sound is. And on this graph, it's on the y-axis and measured in decibels. So because buzz pollination is quite a complex behavior with many different components, it's likely to be impacted by the health of the bee. And this is in itself determined by various stresses in the environment, such as pesticides. And I don't need to give much introduction to neonicotinoids, but just briefly, they are still the most widely used insecticides in the world, and they're routinely used on all major crops, including cereals and orchard fruits and soft fruits as well. And as we heard from Nick, there is a large body of evidence suggesting that neonics have been contributing to the decline in bees and other pollinators. And yet just the moratorium is still underway, and it's due to be reviewed next year in early 2017. So the aim of this particular research is to look at the impacts of these chemicals on buzz pollination behaviour in bumblebees. And in particular, we wanted to look at impacts on the learning of the bumblebees and their ability to improve their buzz pollination over time. So on to the methods. We took a commercial bumblebee colony, Bombus terrestris, and we split the workers from this colony into 3 different groups. And these became our 3 treatment groups, exposed to different concentrations of the neonic thiamethoxam. The low treatment group received 2 micrograms thiamethoxam per kilogram of sugar water, which is equivalent of 2 parts per billion. The high treatment group received 10 parts per billion, and the control group obviously received just sugar water. We chose these concentrations to reflect field-realistic doses. So a bee foraging in an agricultural landscape is likely to encounter these concentrations either in the nectar or the pollen of crop plants or wildflowers growing in agricultural field margins. So these groups of workers happily established microcolonies, and they went through a 9-day preconditioning period during which they had access to a flight arena. So the microcolonies in a box within this cardboard box on the left, and there's a plastic tube connecting them to a large wooden flight arena. And this was simply to give them practice of going in and out of the colony. And after the 9 days, our buzz pollination trials began. So the 3 colonies were connected to the flight arena sequentially for a total of 5 weeks. And when a microcolony was attached, bees were individually allowed into the arena for a total of 10 minutes. And if a bee successfully entered the arena, she was individually tagged with a numbered disc, because she showed herself to be a potential forager. And at the beginning of each trial, so for each buzz pollination trial, 3 flowers were attached to a stick. These flowers are from the plant Solanum rostratum. The common name is buffalo burr, and it's native to Central and North America. It's a buzz-pollinated plant, and it's a relative of the tomato. We recorded the entire buzz pollination trial from the other side of a mesh window with a microphone. So just to give a summary of the data that we collected. Firstly, from observing the trials, we took the arrival and departure times for each floral visit. And then from later analysis in the sound analysis program, Audacity, we took the peak frequency of the first 5 feeding sonication buzzes and the first 5 flight buzzes. And we also took the amplitude of these same buzzes, feeding and flight. And we also took a measure of buzzing effort, so the number and duration of buzzes during the trial. We also measured the amount of pollen collected, so the pollen from one of the bee's legs after a successful buzz pollination trip was collected and stored in ethanol to be later counted with a hemocytometer. And finally, we repeated the entire experiment with a second colony. So on to the results. We spent over 500 hours observing 72 foraging bees, and they carried out a total of 463 arena visits. And a total of 39 bees completed 10 arena visits. And this is the number that we're interested in, the number we were aiming for, because we wanted to get an idea of learning and how bees change their behavior over time. And from these observations, we found that buzz pollination is an innate behavior, as all naive bees were able to buzz pollinate on their first visit to a flower. But it does have a learned component, because experienced bees collected more pollen. But this latter result was dependent on treatment. So the control bees, the non-exposed bees, are shown by the blue line here. And as you can see, they're collecting more pollen over the course of the experiment. The 2 parts per billion bees also learn to collect more pollen, but to a lesser extent. And the 10 parts per billion exposed bees are not learning to collect more pollen over time. And in fact, this is the results for colony 1. By the end of the experiment, the control bees were collecting approximately 60% more pollen than the exposed bees. And we found the same result for the second colony. Again, the control colonies are the blue line here. The difference is slightly less at the end of the trials. For the second, it's about 25% more pollen they're collecting than the 2 parts per billion bees. But again, they're collecting about 60% more pollen compared to the control bees at the end of the experiment. And so these are preliminary results because the analysis is still ongoing. We've got a lot of sound files to analyse. They're proving quite time-consuming to get through. But we are hoping, with all the data we've collected, to get to the bottom of the mechanism behind this effect. And initial results are showing some interesting findings. This graph here is the peak frequency of the feeding buzzes for the first colony. And you can see that the frequency declines with increasing experience. The control bees in this graph are the red, rather confusingly, the red line here at the bottom. So they are buzzing at a lower frequency than the treated bees, but in initial analysis this was not coming out as significant. So what can we conclude from this experiment so far? Buzz pollination is an innate behaviour, but it can be fine-tuned with increasing experience. Field realistic doses of the neonic thiamethoxam disrupt the ability of bees to learn to improve their buzz pollination and collect more pollen over time. And this does have consequences for the fitness of bumblebee colonies, because if workers are bringing less pollen back to their hives, that will have consequences for the successful growth and reproduction of that colony, as other studies have shown. And as I said, we've got plenty of sound analysis to do, so we're hoping to uncover many more details about this fascinating pollination behaviour, about which so many questions remain unanswered. And that's it. Thank you for listening.
Thank you very much. So we have it. Yeah.
So when people like Richard Gill first started exposing these colonies back in 2010, they found that workers on some of the treatments seemed to show increased activity.
Mm-hmm.
So in your hive treatment, they seemed to collect more pollen as naive bees, but didn't get any better. Do you think maybe increased kind of activity they might have been, were they more
Yeah.
active?
Possibly. There was quite a marked difference between the two colonies, and I think this shows that each sort of colony genetic line will react quite differently. And the second colony seemed to show that effect more clearly. Initially, it was the treated bees that seemed to be a lot more active. They were sending out a lot more foragers, and so that could possibly be effect. But we didn't see this so much in the first colony. It was actually the control bees were the workers seem to be working better all along. So I think it depends on the genetics, I think, of the bees themselves. But it's a possible effect, yes.
Any more questions?
I'm just wondering if you could tell me how widespread neonicotinoid use still is, and if it's excluded as part of any agro-environment or HOS schemes?
I couldn't tell you the latter part of that question, I'm afraid. I don't know about the AES schemes and neonics, but it is still used quite widely. They're used on winter wheat, for example, so on arable crops there's still quite high usage. And although bees will not directly be exposed to this, because obviously there's no flowers for them, there has been shown that it does persist in the soil and will run off through into sort of the aquatic system, and that's how it seems to be affecting wildflowers and field martins. And there are other types of neonics, so thiacloprid is used on soft fruits still quite extensively, and on less so recently, but you can buy it for your garden. And then there's other treatments like pet flea treatments are all neonic based, so there's quite high usage still, yes.
So you alluded, I've got sort of just one quick question. You alluded to just genetic diversity possibly being a cause of some of the variation. Do you know how much in those colonies, those commercial colonies, do you know how much genetic variation there is between?
No, well, as they're commercial colonies, they're likely to be fairly closely related because there's quite a high inbreeding in there of sort of bumblebee factories. But no, we don't know, basically. I'm not sure how closely related our two were.
Okay,
No.
well, thank you.
Okay.
And thank you very much. I think we'll move on to the next speaker now. Oh, sorry. That's the wrong one. The right one. Ah. So our next speaker is Virginie Bobo, and she's going to talk to us about agri-environment schemes.
Okay, hello everybody, and thanks for attending this session. So yeah, I'm going to present some results and talk about the effectiveness of agri-environmental schemes on visitor abundance and diversity in apple orchards. So this is a project which started in 2015 and is going on for another 2 years. It's called EcoFruit, and it's about managing ecosystem services for fruit production in different European climates. So just a brief overview of AES. We heard quite a lot of nice talks yesterday about that, so I'll just mention that this is a European measure to promote biodiversity in agricultural landscape, and they take the form of payments to farmers for the creation or maintenance of certain environmental measures. These measures take different forms depending on which country you are, but they include organic or integrated farming, landscape features such as hedgerows, and flooring strip, for example. So these are the ones that we are looking at during, in our study. Are they important? So there was some mixed results yesterday shown, but basically they constitute 22% of the agricultural area in the EU, which is quite a lot, and they take up to 5% of the common agricultural policy budget, which is a huge amount of money. In terms of ecological impact, and especially looking at flower visitors, it was recently shown that there is by meta-analysis that there is an overall positive effect of AES on visitor diversity and abundance, and that this effect was highest at intermediate landscape complexity, so where you have between 1 and 20% of semi-natural habitats within 1 kilometer. These are the references if you're interested in more detail. So what we are looking at in this project is basically looking if there is an increase in diversity and abundance of visitors when there are more than just one AES implemented, and if there is an effect whether, oh no, sorry. So for instance, we are looking here, so is this, yeah. So this here is for conventional plantation, and these are for organic apple orchards. And these ones represent like semi-natural habitat or flooring strip. Initially, we wanted to actually look at flooring strip specifically, but in the 3 areas where we're working on, this was not very widespread, or there were none at all. So we actually moved on to take into account hedgerows and semi-natural grasslands. But anyway, here this is for representing semi-natural habitats, and you can imagine that you would have visitor abundance and diversity relatively low when you have no AES, and when it moves up a little bit when you have one AES, so semi-natural habitats or organic plantation, and you could have an increase even better when you have 2 of these management practices. This increase could be additional, could be synergistic, but like you can imagine it could increase. So that's our initial idea. And then we also looked at whether this changes with this intermediate landscape hypothesis, complexity landscape hypothesis as well. So we can think that if you have a very low diversity landscape like this one, where it's basically only crops, only crops, then you would not go to a very high diversity or abundance of visitors. But if you have a very complex or a more complex landscape, this would increase more. So this is just intermediate complex theory. So going on to the setup, so this is a European project involving 3 labs in Germany, Spain, and Sweden. So we did the sampling in the south of Germany near Constance, and towards Barcelona in Spain, and in the south of Sweden there. Each country had a target of 28 sites, 14 of which should be organic, 14 should be conventional. And within each of these organic and conventional, half of them should have a semi-natural habitat within 50 meters of the edge of a plantation, and 7 should not have any such habitats. They, of course, had to follow a perfect gradient of forest percentage within 1 kilometer. And in the end, it actually worked quite well. So we ended up having 100 orchards, 57 being organic, 43 being conventional. We did manage to find an approximate equal number of sites with and without. So it's relatively balanced. And we have a forest gradient which is very different from country to country. Especially Spain has from 0 to 8% of forest within 1 kilometer, while Germany and Sweden have a more widespread gradient. So now within each of the sites we have several data collection points. So we had target trees here, and so this is about 20 meters from the edge of a plantation, which is located here. So 5 target trees were located here, and then we set up 2 transects. One at the border of a plantation, so it's about 20 meters, and then one inside the plantation, another 20 meters a bit further. And then from the point here, we looked at 50-meter radius around where we were standing, and looked whether there were any areas which were qualified as AES, this being, so ideally, which should have been flowering sweep, but these are now semi-natural habitats. So here you have meadow planted with some trees. So what kind of data did we collect? So at the target tree, we monitored flower count and fruit counts throughout the production period. I'm not going to present the fruit counts here. And then in the transect, we did visitor observations. This is done 3 times a day at the peak flowering day, and then done for 5 minutes each time. So we have a 30-minute observation per site. And we collected visitor diversity and abundance. We also recorded vegetations by using quadrats of 1 square meter, and we had 3 quadrats in the outside transect and 3 quadrats inside transect, where we got the vegetation diversity and the cover for each species. We did the same vegetation surveys in the semi-natural habitat when there was one, and when there were no semi-natural habitat, we used field margin, so this would be something like that. And we also recorded flowering plant diversity and cover. We have also data for each site around the landscape data around each site. So this is 1 kilometer diameter, and we have the percentage of forest cover, the percentage of orchards. We also have a percentage of bee-friendly habitats, which is estimated country-wise. So this does not cover the same type of habitats from country to country. We have a diversity of a landscape, land uses. Sorry, and we have a distance to the closest bee-friendly habitat in there. To analyze the data, we used linear mixed effect models and used country and variety as a random effect. So now going on to the results to present you a little bit what's going on at the by visitor diversity and abundance level. So we recorded between 20 species and 1,400 individuals in Sweden to 37 species and more than 3,300 individuals in Spain. And you can actually notice a slight gradient from the north to the south, where you have more species and more individuals. So that's species diversity and abundance. You can see the blue area here represent Apis mellifera, so between 80 and 90% of all observations were actually from Apis mellifera. And then you have quite a few species. So since we're interested in the impact of AES on the visitors, we're not really so much interested in Apis mellifera. So I'm showing you now what it looks like when you remove Apis mellifera. So here, first thing, don't trust the colors, because, for instance, here this is not Bombus terrestris, but this is Bombus terrestris. So sorry for that. So for instance, in Germany, the most common species was Bombus terrestris, and Sweden as well. But in Spain, it was Eristalis stenax, the syrphid. And so this is just for you to get an idea of the type of species we found. Notice that in Germany and Sweden, it's mostly bees. So we have Tipula, and then there's Eristalis here, and maybe another one, and Bombilius as well, which are not bees. But basically, it's mostly bees and bumblebees. It's a different story in Spain, where out of the 10 most common species, you actually have quite a few flies, beetles, and syrphids. So 5 species. Now, I did the analysis of the visitor diversity and abundance, only with wild species. And we're going to look at the visitation rate, visitor abundance, and visitor richness. And these are the variables that were included in the model. What I was really interested in was the interaction between management and semi-natural habitats, so with AES type within 50 meters of our plantations, and then the same variables on their own. But here you have a landscape variable, orchard, forest, bee habitat, the distance to bee habitat, the landscape diversity, and this is a vegetation diversity and cover that you, yeah. So looking at visitation rate, so this is coefficient plots. It represents the coefficients from the model. So this is the actual like coefficient value, and this is confidence interval. So if it crosses the zero line, which you see in faint gray here, it means it's not significant, when it doesn't, it is significant. So here you see that out of the ones which I was really interested in, management, the interaction between management and semi-natural habitat is not significant. Management is actually significant, with the visitation rate being higher in organic plantations than in conventional ones. And semi-natural habitat does not appear to be significant. It's borderline, but it's not significant. So other variable which seems to play a role in visitation rate are actually the percentage of forest, which has a negative impact on visitation rate, the distance to be habitat, which also has a negative impact, and this is, yeah, it can be understood. And then the flowering plant diversity, which has a positive impact. So flowering plant diversity is actually flowering plant diversity within the orchard, but also outside in AES and non-AES plots. Moving on to visitor abundance, this is an even bleaker picture when you look at AES. So the interaction, again, is not significant. Semi-natural habitats on itself and management practices are not either. What appears is distance to be habitat and the flowering plant cover this time, which both have a negative effect on the visitor abundance. And similar, so this is a typical good example for negative results. So visitor richness also doesn't have an impact on, the interaction of management and semi-natural habitat doesn't have an impact on visitor richness, and same for these two here. So this is really preliminary data, and what the data that we included is actually for the AES, so the semi-natural habitat close to the plantations, is actually a yes or no. So if there is an AES or if there is a semi-natural habitat, we say just yes and otherwise no. So it's a very crude measure of that. And I actually wanted to see a bit the raw data. So this is for visitor abundance. That was this one here, which is the one which is closest to significance. Well, anyway. So if you look at this graph, you have conventional and on the left-hand side and organic on the right-hand side of each box. And the left box is for when there's no semi-natural habitat. The right one is for when there is semi-natural habitat. And what you can see is there's a trend when you actually have semi-natural habitats, you increase the number of the abundance of visitors. So this is actually going in the direction which I thought we could go. Like here you have no AES, you have one AES here, one AES here, so it's slightly higher, and then an additive effect when you have two AES. But it's not significant. What I think we need to do is actually go a little bit more into details, because at the moment we can't say that we can actually say that there's no effect of semi-natural habitat, but there's no synergistic effect of AES. And I also didn't show you the data, but there's no intermediate, like say, complexity going on there. So, but I think we have a possibility to actually look at the size and numbers of the semi-natural habitats within 50 meters and get, instead of having a categorical variable which is quite crude, so having something a little bit more detailed, and then also the quality assessment. And okay, I'm going to finish here. And then look at also within country analysis, because this is really international level. So with that, I want to thank you, and these are the people who are participating in this study. And yeah.
Two quick questions, if they're quick. Is it quick?
Yeah, it's good, yeah. Maybe I missed it, but your visitation rates and your abundance, did decay also by using honeybees?
No,
or is
no.
everything except honeybees?
Yeah, this is everything except only this. Yeah.
And did you look at each country separately?
So I did that, but this is even more preliminary data. You have different pictures for each country, actually. And in Spain, it's quite interesting because AES actually, or the presence of semi-natural habitats actually has an effect. And they have landscapes which are a lot more, less diverse than what we have in Germany or in Sweden. So I guess that is probably why. So, yeah.
I'm sorry, hello, thank you for that talk. I didn't hear that question, so maybe this is the same question. I don't know.
Well, shoot out.
Do you think that the effect you expected on the wild visitor fauna from Mars by all those
No, because the honeybees are not taken into account in the
So
analysis.
you're only looking at a community of wild visitors, which
Yeah.
is in the context of dominance by honeybees.
Yeah, yeah, exactly. Yeah, I mean,
That's
there
why
could
you
be
don't see the patterns.
there could be interactions between honey, like at the and in the plantation. But I mean, there were so few bees when we did the observations that I don't think they could disturb each other much, at least. But yeah, it is possible, and I'm also going to run the analysis for that.
Thank you very much.
Thanks.
So hopefully.
It's working.
So our next speaker is Jürgen, and he's going to be
And my surname?
Schreper,
Yeah, excellent.
and he's going to be talking about promoting wild bees in agricultural landscapes.
Okay, thanks for this nice introduction. I'm going to give an overview of my PhD thesis that I finished about a year ago, supervised by David Klein. And it was or is entitled Promoting Wild Bees in European Agricultural Landscapes, the Role of Floral Resources in Driving and Mitigating Wild Bee Decline. And I'm afraid there will be some overlap with the previous talks, but anyway, let's just continue. So, in addition to the well-known honeybee, we have a lot of wild bee species in Europe, about 2,000 of them, and already here's the gradient Virginie talked about. You have very few bees in the north, only 3 in Iceland, and many bee species in the southern part of Europe. And also already addressed in the previous talks, bees are important for pollination, but also represent an intrinsic value for biodiversity. But many of these wild bees have been declining over the last couple of decades. And this graph, probably many of you will know it, the study by Koos Biesmeijer, I should know how to pronounce that, Koos Biesmeijer and colleagues, published in Science. This shows the change in bee species richness in the Netherlands in grid cells, and they compared the period before and after 1980. And red is bad news, so as you can see through your eye laces, the Netherlands is turning rather red, which means bad news for bees. Especially agricultural landscapes have become very impoverished when it comes to bee communities. And currently, more than half of the bee species that we have in the Netherlands, about 350, are on the red list. So there are several drivers associated with this decline. Nick and Penelope already addressed the pesticides. Bees can also suffer from invasive species, pathogens and parasites, climate change, but general consensus is that land use change and agricultural intensification since the second half of the previous century is one of the most important drivers of the decline of many bee species. And these graphs strikingly show what has happened in the Netherlands over the past 100 years or so. Here you see the area of open semi-natural habitat, which is the typical preferred habitat for bees, extensive grassland, heathland, etc. And you can see it has declined very sharply, and currently only 20% of what was once there is still remaining. But this is not the only thing that's going on. Also, existing farmland has become much more intensively used. You see a graph of the use of nitrogen in farming, and nitrogen applied per hectare is generally associated with other measures of intensity like mechanical disturbance, use of pesticides, herbicides, mowing and grazing frequency. And together these two factors result in the loss, fragmentation and degradation of habitat. So it's generally assumed that these drivers, land use change and habitat agricultural intensification, mainly impact bees through the loss of their floral resources. But this has never been tested. So what we did was we went into museums and collected pollen. We scraped them off the legs and stomachs of bees, and we analyzed the pollen content of the plant species composition in pollen loads of different bee species. And we calculated a weighted mean population trend of the plants in the diet of bees, and we related that to the change in the bee population. And this period compares the period before 1950 and from 1975 until more or less 2000. And as you can see, as the preferred plants in a bee's diets decline, so do the bees that depend on them. On the other hand, if plants are doing okay, the bees that like to forage on them are also doing quite okay. So another thing that we found was that as bees are larger, they are more prone to decline. And we can explain this by the fact that larger bees generally require larger quantities of pollen. And so if in the agricultural landscapes floral resources decline, these larger bees are getting in trouble sooner. So together these results point to loss of floral resources as a main driver of bee decline. So the question pops up, what can we do about this to mitigate bee decline? Well, it seems obvious, enhance floral resources. And one way to do this is by the use of agro-environment schemes. Virginie explained what it is. You have different types. And these can either directly or indirectly, by extensifying management, result in enhancing floral resources. But the general effectiveness of such measures have been debated, especially from the 90s onwards. And it seems that the effectiveness of these measures varies a lot between landscapes, regions, between types of measures, but also within measures. So if you want to mitigate bee decline, you need to know where this variation comes from. So what we did was we carried out a meta-analysis, compiled the results of 71 studies that compared bee richness and abundance in agro-environment scheme sites and in conventional field margins or conventional control sites. And we looked at the effect of landscape context, already discussed in a previous talk, but just quickly. What we found was that over here you have the effect size, so if it doesn't cross 0, it's a positive significant effect. We found that the effectiveness of measures is highest in simple landscapes. In cleared and complex landscapes, effectiveness is lower. And this can be explained by the fact that in cleared landscape you have hardly any semi-natural habitat left. So you do not have a large bee species pool that can respond to the implementation of your measures. On the other hand, in very complex landscapes, you have a lot of semi-natural habitat, a large bee species pool which continuously spills over in the agricultural matrix, and consequently biodiversity is already high everywhere. So if you implement a measure, it doesn't really add anything. So it is in the intermediate situation where you still have some source populations left, but biodiversity isn't already high everywhere, where measures are most effective. But of course, it's not only the landscape, it's also the extent to which measures, really, okay, the extent to which measures improve local conditions compared to control sites. And this is what we looked at over here. I'm going to quickly, if measures improve floral resources more compared to the baseline, they are more effective. Makes sense, right? So it seems logical that directly enhancing flower resources by sowing flowers is particularly effective. And this is exactly what we found in a large-scale field study. In 4 countries, we used a Becky design, and here you have the control field margins for bumblebees and solitary bees, and in red you have the wildflower strips. And in all countries, after 1 or 2 years, these flower strips result in higher densities, higher richness of bees. And they even result in more red-listed species. And it's a common critique that agro-environment schemes only benefit common widespread species. So, really great, good news, or not? Because the evaluation of these measures is mostly done by carrying out transect surveys. So you're catching, counting bees, and you're actually measuring foraging responses. So if we consider this hypothetical landscape, each dot is a bee, and what happens if you establish a wildflower strip? You find more bees. But is it because all bees from the surrounding landscape aggregate on these very attractive patches of flowers? Or does it also result in actual benefits for reproduction and enhanced populations? You cannot tell from these transects. So what we also did, we monitored reproductive output in trap nests. Okay. And what we found was that it's a bit disappointing. Here you have result for Hilaeus species, and if you see over the years, again in red, it's the wildflower strip, there's no effect at all on the brood cell production of this bee species. But it makes sense because this bee species mainly forages on bramble, and farmers do not really like sowing bramble in their fields, so we didn't put it in our wildflower strips. But we did put yellow Asteraceae, Tanacetum vulgarum, and this bee species really is fond of Tanacetum vulgarum. And here you see that after the second year, these wildflower strips result in actual benefits for reproduction and larger populations of the bee species, and that should ultimately be the goal. So in conclusion, the loss of floral resources is one of the main drivers of bee decline, and you can quite easily enhance local densities of foraging bees by sowing flowers. But if you really want to enhance populations, you should sow the specific host plants of your target bee species. So thanks for your attention.
Thank you very much. You're on time, it's fine. You have plenty of time. Can I ask you if you have any questions? Okay, well, I'll start with the question. So going back to your herbarium study, when you were looking at pollen that you were collecting from the bees, do you have any idea about how much that represented the flowers that they actually forage from? Or how, because presumably you must have lost quite a lot of pollen in the whole process of getting them into the herbarium.
Well, it's it's about 20% of all bees in entomological collections still have pollen loads on their bodies.
Right.
And we sampled the bees that were caught before 1950. So we assumed that before 1950, the landscape was more or less intact, and bees actually still had a choice. So it's an assumption, but we are pretty sure that it reflects the actual preference of these bee species. If you go out in the field and sample them now,
Yeah.
you don't know if it's really the preferred resource or just there's nothing better left.
Yeah.
So, yeah.
Okay, thank you.
Okay.
Does anybody have any other questions? Yes, please.
Hi, it's a very interesting talk. I went to Rowntree yesterday about using the environments and wildflower strips and how they was working on some bumblebees with solitary bees. I wondered if the European agri-environment schemes had different performances between countries, if they were more targeted towards other species other than bumblebees.
Good question. Indeed, in the UK, you pretty much have these pollen and nectar mixes, right, with Fabergé for the bumblebees. I must admit that in the Netherlands, we do not even have an agri-environment scheme that includes sowing wildflowers. It's being done more and more, but mainly by public initiative. What we did in the field study was sow a mixture targeted at bumblebees as well as solitary bees, so also the Asteraceae, Malva, a lot of species, and we used the same seed mixture in all 4 countries. So, does that answer your question? Okay.
Any more questions? Is it a quick question?
I was just wondering if you're going to try and use your research now to try and influence policy, to try and then get areas to actually match their planting schemes with the species that are local and need kind of increasing, if you're seeing a match between them.
That would be, that would be nice, but I'm not currently involved in that. It's a good suggestion. Yeah.
Okay, well, thank you very much. And we'll move on to the next speaker now. Okay. Our next speaker is Julianne Horn, and she's going to talk to us about using modelling frameworks to assess the effectiveness of ecological focus areas.
Thank you very much for your nice introduction and welcome to my talk. Yes, important drivers for the increasing colony losses of honeybees are, of course, parasites and diseases and also pesticides, but really important are also land use changes that lead to simplified cropping patterns and also to decline in semi-natural habitats at large landscape scales. And so we have many agricultural systems, temporal lags and nectar and pollen supply, especially in early spring and in the summer months. The EU has already implemented some mitigation measures to improve the environment, and in the current Common Agricultural Policy reform of the EU, there are currently implemented the ecological focus areas that have to be implemented on at least 5% of the agricultural area, but there are critical voices that these current obligations are set too low, are too broadly defined, and that the farmers are able to meet these current obligations with their current practices without changing anything. So that's why I want to demonstrate today this usability of modeling for evaluating the effectiveness of such ecological focus areas for honeybee colony performance. And I simulated how the temporal and spatial continuity in nectar and pollen availability at the landscape scale is affected by the crop diversity and the implementation of different ecological focus area types and coverages, and also their spatial configuration. And so we are asking which ecological focus area types and coverages have the highest potential to meet the honeybee colony's food requirements and to ensure the long-term viability of honeybee colonies. To do this, we developed a modeling framework in a two-step approach. At first, to design the ecological focus area scenarios, we developed agricultural landscape generator. And here we and here we implemented a simple structured farmland and different baseline farmlands of different crop compositions of low and high diversity. And also we define here the ecological focus area types and coverages on the landscape, agricultural landscape area, and also their spatial configurations. And to get such daily nectar and pollen flow maps as input for the next modeling step, we implemented here the flow data of crop species and the plant species for the ecological focus area types, and calculated the pollen and nectar flow on a daily basis over the whole year related to the crop field or crop field size or the size of the ecological focus area field or margin size. And here we considered and inputted the data about the flowering period, the flower density, nectar and pollen amounts, and also the sugar concentrations. Yes, in the second step, the ecological focus area scenarios are implemented and analyzed in an existing honeybee simulation model. This is called Beehive. I will tell you 2 minutes later something about this. We used to design the ecological focus area scenarios as simple structured landscape of a typical heavily managed agricultural landscape with few and really large patches. And here we implemented 2 different crop compositions, one of low diversity consisting of the most typical European crop types and one of higher diversity consisting of 8 typical European crop types. For the baseline farmlands, no ecological focus area elements are implemented. And to implement the ecological focus areas in terms of their coverages, we focused on current and planned obligations of the Common Agriculture Policy Reform. So we implemented the 5% of the currently reform and also 7% that is planned for the next period for 2020. And also we considered claims by a conservation organization that claims that the areas should increase to 10%. As types of ecological focus areas, we implemented set-asides, nitrogen fixing crops, field margins, for example, flower strip margins and grass margins for the buffer strips, and also hedgerows. And these types are these which are frequently chosen by the EU member states. And for each combination of ecological focus area type and coverage, we analyzed 10 different spatial configurations. The outcomes of the ecological focus area scenarios we analyzed in the model BEHAVE, which integrates a colony module that describes the colony structure and dynamics, which are influenced by the colony stores and also by specific mortalities of the different life stages. And this model also integrates a virus transmitting varroa mite population, which is linked to the colony module, and also a foraging module. Here, foragers can collect nectar and pollen from a landscape, and this foraging module is linked via the energy income to the colony module, which determines the colony's pollen and honey stores, and also the structure of the colony and the dynamics. With this model, we calculated for all these scenarios a point-to-worker productivity index, and this point-to-worker productivity index is composed of the number of worker bees that were ever produced in the colony over the simulation time of 5 years. And this is multiplied by the average pollen consumption of a single worker bee over its complete lifespan. And we can compare these point-to-worker productivity index with empirical estimations of a viable honeybee colony, which produces 100,000 worker bees per year and which requires 20 kilogram pollen per year. Yes, in this figure you can see the point-to-worker productivity values for the different ecological focus area types, the different coverages, and also the different spatial configurations. You see that most of the ecological focus area types are similar to the baseline scenario. For example, if we implement bean cultures as nitrogen fixing crops or also grass margins, there is no change at all considering this point-to-worker productivity. The red dashed line indicates these thresholds, these empirical thresholds of the pollen requirements of a viable honeybee colony over the simulation time. And you see we have only one ecological focus area type which slightly exceeded this threshold. This is the flower strip margins, but you see we have a high variation among the species configuration for 3% coverage, and which is lower for 10% coverage. But regarding that, these all these coverages only slightly exceed this threshold, it's really unlikely that in a cropping system of low diversity, this ecological focus area type will really improve the honeybee colony under realistic conditions. If we have a cropping system of higher diversity, you can see that we have some spatial configuration of, for example, clover culture implemented as nitrogen fixing crop and also Phacelia set-aside that exceeded this empirical threshold of point worker productivity. And because we have here some fields that are located really close to the hive. And also you can see that the flower strip margins from 5% onwards, the dependency from the spatial configuration is exceeded by the coverage. So in this figure you can see the nectar and pollen supply over the season, and here you see it for the implementation of 10% nitrogen fixing crop clover in the agricultural system of high diversity, and you see you have continuous supply of nectar and pollen until late summer, but after this nectar and pollen supply you have a late seasonal gap in this nectar and pollen supply. So that's why that is important that the clover fields are located close to the hive to get sufficient energy income for sufficient honey stores for overwintering. For the flower strip margins, if you implement 10% of flower strip margins on the agricultural landscape in cropping system of high diversity, you have a continuous forage supply over the whole season. Okay, yes, honeybee colony performance and thus viability is influenced by the forage availability in the landscape, and this is influenced by, at the one hand, by the crop diversity, but mostly by the semi-natural frequency and location of the semi-natural habitats, but also by the implementation of ecological focus areas, coverages and types, and also the spatial structure. And all this determines the timing and frequency and also the duration of forage gaps where no nectar and pollen is available to the bees, and this determines the range of cascading effects on the mortalities, on life stages, and also on the force of worker bees for nursing and foraging tasks. Okay, yes, to conclude, such modeling frameworks are important to maximize the benefits from ecological implementations and to improve our understanding about the impacts of the extent, spatial configuration and quality of ecological focus areas in tune with the needs and behavior of single species and at least on species communities. Did I have time? Finish. Okay. Then I finish.
No, you can carry on if you want.
Thanks.
Thank you very much. So I'm not being very clear. There is a bit of time for one or two questions if people have questions. Yes, please.
Hi. Do you know how you showed the kind of total demand from a honeybee colony over the year for their food supply?
Yeah.
Pollen supply. Do you have data on how that changes through the year rather than just the total? I'm assuming it really crashes very quickly at the end.
Yeah, there are only some empirical estimates. There are not so much data available at the moment. So this 20 kilogram pollen is over the whole year.
Whole year, so you don't know
Yes.
how that percent changes.
Yes.
I think we'll move on now. Thank you ever so much for your talk. So our next speaker is Johanna Häusler, and she's going to be talking about using spatially explicit modelling to quantify bee dynamics, wild bee dynamics.
Thank you for the introduction. I will continue with the modeling approach. And yeah, I forgot to put on the no Twitter sign, but it's unpublished data, so please do not Twitter or tweet any results graphs. So yeah, I will introduce you to a spatially explicit process-based modeling approach to quantify wild bee dynamics and their pollination services in agricultural landscapes, which my co-workers and me developed at the Center for Environmental and Climate Research in Lund University in Sweden. Yeah, I mean, I will just go through this very quickly. We heard a lot about it already. Wild bees are important pollinators, but they're threatened and are declining due to land use change, land use intensification, and this also impacts the pollination services they contribute to wildflowers as well as crops. So what can we do to improve the situation for pollinators in agricultural landscapes is to implement pollinator-friendly habitats such as field margins or flower strips offering high amount of resources such as floral or nesting resources. Why are models useful tools to do this? Empirical studies often are restricted by time or the scale, the landscape scale, what they can study. So landscape spatially explicit process-based models are useful tools to study effects over time and landscape scale-wide. So we want to answer questions such as how does the landscape structure influence pollinator population dynamics and the pollination services they provide, and how much pollinator habitat is needed and how it should be distributed within agricultural landscapes. So now we'll quickly introduce you to the pollinator model we developed, which you can see here. So as model input, we have historical land use maps from 2007 to 2013 from southernmost Sweden, the Scania region, which is a heavily agricultural area dominated by mass flowering crops, for example, oilseed rape. So from these historical land use maps, we derived GIS rasters and then selected landscapes of 10 by 10 kilometers with a cell size of 25 by 25 meters. In this exemplary land use map, which you can see here, each of different colors describes a different land use category, which then has assigned a specific land use category, nesting quality, floral coverage, and attractiveness factor. These are based on expert opinion and published literature. And so with oilseed rape being the most dominant crop, mass flowering crop in Scania, I'll show you it as an example. For example, here, and we also divided our season dependent on oilseed rape. So we have flower period 1 during oilseed rape flowering, and then we have flower period 2 after oilseed rape flowering. So the first floral coverage, for example, we have a very high floral coverage of oilseed rape at the beginning of the season, but a very low floral coverage at the end of the season, and also very high attractiveness for bumblebees at the beginning of the season, but a very low attractiveness at the end of the season. And in oilseed rape, we assume that nesting quality is quite low. So now we'll quickly go through the pollination model itself, what's going in there. So here you can see a simplified two-stage seasonal model of common early active bumblebees, which you can see here. They are very important pollinators in the landscapes that we look at. And so they are foraging so at the beginning of the season, the queens start to forage when oilseed rape is flowering. And then during this period, they start to produce workers. And then after the flowering peak of oilseed rape, a subset of workers starts to forage. And then the colony is growing. And based on this amount of resources that the workers have foraged in this period, the queens start to produce new queens and males, which we, however, neglect in our model. Then at the end of the season, the new queens disperse to overwinter, and the workers' old queens and males die. Then during the winter, we have, of course, mortality, and at the beginning of the new season, after hibernation, and the queens start to emerge. Yeah, so the bumblebees we looked at now are all central place foragers with quite large foraging distances and long periods of activity. And because they have such a long period of activity, the flower availability between this long period of activity is of quite importance, as we heard before already. So, as model output, we have spatially explicit visitation rates, both to crops as well as wildflowers. Yeah, so of course there have been previous models quantifying pollination ecosystem services, for example by Eric Lonsdorff and colleagues, and which estimate pollination ecosystem services using distance from likely nesting sites, but those, however, ignore resource-dependent optimal foraging and its consequences on population dynamics, which have been observed, however, in empirical studies. And also, they only provide relative measures of pollinator visitation to crops. Then there have more recent models been, for example, by Olaf Ulsen in 2015, which integrate optimum foraging, or by Crone and Williams in 2016, which consider population growth within and between years, but they don't combine these two processes. So with our model, we combine these two processes by integrating preferential use of more rewarding floral and nesting resources, which you can see here. So here you can see on the x-axis the distance from nest sites, and on the y-axis the percent of foragers. And the different colors depict different floral values. So in our modeling approach, or in former modeling approach, we see that there's like a, when there's no difference between floral values across the landscapes, that there's like a decline. But when we have resource-dependent foraging, so dependent on the quality of the resource, that with the distance, the percent of foragers can change. Then, furthermore, we allowed for different movement distances for foraging and queen dispersal. So for now, we had as input values based on published data. For mean dispersal distance for foraging, we had 530 meters, and for mean dispersal distance when flying to nesting sites, we had 1 kilometer. Then we also consider resource-dependent population growth within and between years, which you can see here. So we have, as I showed you before, we have growth during the season, so the colony growth, and growth at the end of the season when the population size is actually growing in terms of number of colonies. So here you can see on the x-axis the resources collected during foraging, and on the y-axis the number growth in number of bees. So you can see here is our growth function, which is a function of a log normal distribution. And so you see here, dependent on resources foraged at the beginning of the season by the queens when they started nest initiation, is the growth of the workers during the season, up to a maximum number of workers, which is based on literature data. And then at the end of the season, based on the amount of resources the workers forage at the end of this period, floral period 2, is the growth rate of the new bumblebee queens. Yeah. Then we also produce spatially explicit absolute flower visitation rates, which then can be used in crop pollination models, whereas previous models only produced relative flower visitation rates. Yeah. Now we'll introduce you to a model application which we did. So as we heard before, it is very important to have pollinator-friendly habitat, such as grassy field margins and or late blooming flower strips in agricultural landscapes. So we wanted to test the effect of implementing these management interventions over time and at different landscapes. So so bees need something to eat and something to sleep in, like we do. So we have flower strips and grassy field margins, and we implemented them in the landscapes. Flower strips were assigned very high values in flower cover values and bumblebee attractiveness. So at the end of the season, because they were late flower strips. And we also based the amount of flower strips in a landscape we looked at, dependent on the amount of agricultural land in this particular landscape. Then we also had field margins, which were just broadened at, like, every field margin in the landscape was broadened with a high amount of nesting, offering on nesting resources, but low amount of floral resources. Yeah, then, as I already said, we also looked at landscapes with different amounts of oilseed rape, fields, and semi-natural habitats, because we were assuming that landscape characteristics also played an important role in how effective management interventions actually are. Yeah, so these are our results. Because we tested we tested it over a longer time frame. So we had 7 consecutive maps of 7 years, 2007 and until 2013. So we repeated these land use maps and thereby mimic crop rotation for 5 cycles. This explains the sort of like repeating pattern here. Because we wanted to see the effect over time, and also if there's a time lag in the effect of an emergence. So I just, and you can see here just the one example of one exemplary landscape. It's not the mean. So for bumblebee colonies, we see that flower strips were 3 times more effective with the effect in increasing bumblebee colonies over time. So we have a much higher effect of flower strips, which you can see here is like the black dotted line, compared to field margins, which is the the gray line. And this is also supported by empirical studies, which have shown that only implementing grassy field margins with no flower resources had actually very little effect on pollinators. We also see, which we've heard before, that actually offering resources when they are very limited, flower resources when they are very limited at the end of the season, has a very high impact on bumblebees. Yeah, we've heard this before. Then we have also see that we have actually very little benefit of a joint scheme when we combine these 2 methods. Then flower visitations. Flower strips were also very effective in increasing early season visitation rates, including oilseed rape visitation. But however, they decrease late season flower visitations. And this is due to competition for resources. So when we implement late season flower strips, they actually draw away bumblebees from the actual flowers that we had in the area before implementing flower strips. This dilution effect is also shown in empirical studies, and has been demonstrated by phytometers by Verena Riedinger or by Andrea Holschuh by European Wild Field Studies. Then we also found a time lag in the emergence of an effect of management interventions. So we see that the effect, which has also been shown empirically by Simon Potts, when the effect of a management intervention is higher after it has been implemented for several times. So on the other hand, however, we did not see an effect of increased cover of oilseed rape on bumblebee productivity, which has also been shown empirically, because this does not, the resources collected when oilseed rape is flowering does not allocate to reproduction of reproductives, but only to workers. So the colony is growing, but that doesn't mean that they actually produce more queens or males. Just quickly, the conclusions. It is important that we identify which resource is the limiting factor in our landscapes. We've seen that even though oilseed rape cover and heterogeneity themselves did not have an effect, we saw that the impact of increasing flower resources was more pronounced in heterogeneous landscapes, and the interaction of resource-dependent foraging, apparent competition between flower resources, and long-term effects highlight the added value our model provides compared to previous modeling approaches. And we also see that our results reflect empirical observations. However, our model allows to explore these observed effects further over an extended time frame and larger landscape scales. And yeah, with our model, we provide a useful tool to improve evidence-based policy by linking pollination ecosystem services to land use. Yeah, so it's ongoing work, hopefully to be published soon in Ecology and Evolution. And yeah, so I want to thank my collaborators at CEC, Lund University, and of course, thank you for listening.
So I think we'll move on from here straight away. If you want to ask any questions, please do, and in the break. Thank you very much. Sorry. Okay, so our next speaker is Panos Theodorou, and he's going to talk to us about insect pollinator diversity and pollination in changing landscapes.
All right, so thank you all for coming here. So today I'm going to discuss with you a study we did to investigate insect pollinator diversity and the ecosystem service of pollination in changing landscapes. So we heard a lot about how important pollinators are and pollination is, so I will skip this slide and concentrate a little bit more on our results. We also heard a little bit about the global change pressures on animal pollinators, and the one that I will concentrate upon is landscape alteration, or in general land use change, let's say. And one of the main drivers of land use change is urbanization, is due to urbanization. We know that the global human population is increasing, and consequently also the urban human population will also increase, causing also an increase in urban land use. In general, we know that urbanization is a major driver of shifts in insect pollinator diversity and pollination, with overall negative effects, but also some studies showed some positive effects, especially for some certain pollinator taxa. So there's a growing interest in the role of cities, not only in requiring, but potentially also in supplying biodiversity and pollination. When we talk about biodiversity, we often refer to species diversity. And consequently, when we work on the effects of land use on biodiversity, we work on the community level of biodiversity. However, as we know, biodiversity encapsulates information across all range of biological organization, from genes, species, and ecosystems, and intraspecific genetic diversity, its fundamental level of biodiversity, is shown to enhance population fitness, affects ecological processes, and also co-varies theoretically with species diversity. So here in our study, we investigated insect pollinator diversity, species richness, phylogenetic diversity, and intraspecific genetic diversity in rural versus urban areas. So to do that, we picked up several urban areas in Central Eastern Germany. So we had sites in 9 main cities in Central Eastern Germany and 9 rural areas in the nearby in a pair design. In each of these sites, we used pun traps to collect flying insects, and we used meta barcoding for our biodiversity assessment. So what we did, we collected flying insects using those pun traps, and we emptied them every day. After collecting those insects, we homogenized them, we extracted DNA from that soup, and then we did a mass PCR amplification of the CA1 barcoding gene, 454 parallel sequencing, and then we used several bioinformatics steps for OTU picking and taxonomic assignment. So this resulted in an OTU table, right, where we calculated species richness of 4 main insect orders, Diptera, Lepidoptera, Coleoptera, and Hymenoptera. And we also calculated 2 phylogenetic species diversity indices, phylogenetic species variability, which measures how phylogenetic relatedness reduces the variability of a hypothetical neutral trait in the community, and phylogenetic species richness, which is actually the phylogenetic species variability multiplied by the OTU richness in each community. So while we were also pun trapping, we also collected individuals from the most abundant pollinators across those 2 ecosystems. And here I will present results on Bombus lapidaris. Hopefully then I get some money and I can do also the same analysis for the other 2. So in each of these sites, in each of these 18 sites, as I mentioned, we also collected Bombus lapidaris workers, and we extracted DNA from them, And then we did Illumina-based rat sequencing, discovering about 20,000 SNPs, and then based on that we calculated the genome-wide diversity, intraspecific genetic diversity, sorry, of Bombus lapidarius population across the two ecosystems. So here are some of the results we got. Overall, we found higher OTU insecta richness in rural versus urban areas. We also consequently found higher phylogenetic species richness in rural versus urban ecosystems, and however, we did not find any difference in phylogenetic species variability between the two ecosystems. Going now in each of the orders, for Lepidoptera, we found higher OTU richness in rural areas, also for Diptera compared to urban ecosystems. We didn't find any difference in Coleoptera. However, for Hymenoptera, we found higher OTU richness in urban versus rural areas. And these are the two main families, let's say, that we also investigated. For Seafish, we did not find any differences between the two ecosystems. And for bees, we found higher OTU richness in urban versus rural ecosystems. For intraspecific genetic diversity of Bombus lapidarius, measured either as heterozygosity, nucleotide diversity, or percentage of polymorphic loci, we did not find any differences between the two ecosystems. So here I also correlated species richness with genetic diversity of Bombus lapidarius, and across both ecosystems and within each ecosystem there is a strong correlation between heterozygosity and insect OTU richness. So what we also wanted to explore here is to identify which are the main drivers of pollination across the urban-rural transition, and whether genetic diversity is an important component of biodiversity that could predict the ecosystem service of pollination. So to do that, we needed to find out, pick up a pollinometer to measure the ecosystem service of pollination. And based on a previous study using different plant species, we found out that red clover is a suitable one. So what we did, we grew red clover in glass houses from seeds, and just before flowering, we packed some flowers, we marked some flowers, and then we put them in the same sites where we conducted our biodiversity assessment, and at the same time, of course. And we also monitored those plants to see what is visiting those plants, and also we characterize the local habitats, such as flower richness estimates and so on, abundance. We also estimated the abundance of co-specific pollen for Trifolium protense in each of these 18 sites. And furthermore, in each of these areas, what I didn't mention before, we also characterized the landscape before picking up the sites that we did. So all sites also had some differences in relation to their landscape composition and landscape configuration at 1 kilometer buffer zone. So we also estimated the proportion of agricultural land, the proportion of green areas, and connectivity of those green areas using a proxy, that of edge density. So here are some of the results regarding seed set and visitation rates to our red clover plants. We found higher mean seed set per plant in urban versus rural areas, higher visitation rates and higher Bombus visitation rates in urban versus rural areas to our experimental pollinometers. So to disentangle local habitats and landscape scale effects and all the facets of biodiversity and how they are correlated with seed set, we use a structural equation modeling approach. Most of the visitation rates to our red clover plants came from Hymenopterans. So we use Hymenopterans OTU richness and Hymenopterans phylogenetic richness or variability as potential predictors of seed set together with Bombus lapidarius intraspecific genetic diversity. So we use structural equation modeling, having some hypothetical links, and then we did a model selection to find out which are the most important ones. So here I start from local scale to landscape scale effects. These results are across both landscapes. The analysis also was done in each landscape separately. So here I am showing the results across the two landscapes, the two ecosystems, sorry. So dashed lines are links that they were hypothesized, but they did not remain in the final model due to the fact that they were decreasing model fit. And so what we found, starting from the local scale, we found a positive effect on local flower richness on Hymenopteran OTU richness across the two ecosystems. We found a positive effect of edge density on Hymenopteran OTU richness and a negative effect of the proportion of agricultural land on Hymenopteran OTU richness. from our estimates of biodiversity, Hymenopteron OTU richness and Hymenopteron phylogenetic species variability had the most higher correlations with the variations in seed set of red clover. Bombus lapidarius, even if it was a positive correlation, it wasn't significant. And here I have to say that the Bombus lapidarius visitation rates were also significantly correlated with seed set, just to keep that in mind. So, to conclude, overall, we found higher insect pollinator richness in rural compared to urban sites. Patterns varied between taxonomic groups. So we found higher Hymenopteron and consequently also higher bee richness in urban versus rural areas, probably due to the availability of lower resources and nesting resources, potential nesting resources in urban areas for that order. We also found positive species and diversity correlations, OTU richness of all insects and genetic diversity of Bombus lapidarius. I have to be cautious here, but this indicates that could be that by conserving species richness, you also conserve genetic diversity. But for that, I need to also work on with the other two main pollinators of those areas, just to have that complete down. We found strong associations also between biodiversity and landscape heterogeneity across ecosystems, pointing out to the value of landscape ecology for studying biodiversity. And also we found that Hymenoptera OTU richness and phylogenetic species variability were better predictors of red clover compared to interspecific genetic diversity. And that is not something that we didn't hypothesize. It's okay, due to the fact that pollination is a product of an interaction, and probably visitation rates are more important than intraspecific genetic diversity in explaining variations in seed set. So with the increasing urbanization and with the proper management of urban areas, I can see that urban areas could be important for pollinator conservation. And with that, I would like to thank many people that helped me to do all this stuff that I presented to you, and also thank you for your attention.
Thank you very much. Does anyone have any questions?
Yes.
So I was interested, you were talking about the different components of biodiversity, and in your structural equation model you had OTU richness and phylogenetic species variability as separate predictors.
Yes.
But they're both different expressions of Hymenopteran biodiversity, and they're both based on the same set of observations. So I wondered, what was the rationale behind us having them as independent predictors?
Well, the rationale is taking phylogenetic diversity as a proxy for functional diversity, of course. It's not correlated directly, or it shouldn't really correlate so much compared to phylogenetic species richness. That index probably correlates very much with Hymenopteron OTU richness. But phylogenetic species variability, it has a historical evolutionary kind of perspective in how communities could, let's say, diverge or not. And in this kind of study, I use it also as a proxy, also as a correlate with phylogenetic diversity. Yeah, I don't know. Is that your question?
Notice in your models you don't have the actual abundance of Hymenoptera or other pollinators.
No.
So is that because you didn't actually count them?
Well,
Okay, so does richness actually just stand in for abundance?
we don't have abundance in our models, and we could measure abundance. We could have, we could have measured maybe from the pantraps before smashing them up and getting their OTU richness from, but we didn't. So we just have richness, and with the meta-barcoding, you could only get down to richness, and you cannot get a proxy based on number of reads or so in terms of abundance.
Which comes to my second half of the question.
Yes.
Which is, without actually counting them, there's a very strong correlation usually between abundance and richness. In a larger sample of bees, you'll find more species of bees. Is there any way to at all tease that apart, or is that just lost?
Well, if we could potentially think that intraspecific genetic diversity is a proxy for abundance, or a proxy of effective population size, and finding out that we find correlations with OTU richness, it points out that maybe we are not missing that much by excluding abundance, I mean, in our measurement of insect diversity. But however, yeah, we didn't do it, so I'm not sure whether that changes anything. Yeah.
Okay, thank you very much. I think we'll move on then. Thank you.
Yep.
So our final speaker is Ulrika Sammersgaard, and she's going to talk to us about pollen limitation and bee abundances.
Thank you. So now we move down to the more tropical part of the world, and I will tell you about findings from the 2 final chapters of my PhD thesis. And let's see. So the aim with these studies were to investigate if and how the local forest cover affect the bee community and the pollination services in an Ethiopian agricultural landscape. And this was part of a larger project with the aim to evaluate ecosystem services and the services coming from moist upper mountain forests. And the background to this project is that we know very little about pollinators in sub-Saharan Africa and even less about their pollination services. But from Europe, we know this one, that increased complexity and heterogeneity in agricultural landscape, and also the presence of semi-natural habitats and natural habitats is generally good for biodiversity and also for pollinators, and that conversion of land into agricultural land and the intensification of land is generally quite bad for biodiversity and also for pollinators. And we know this from especially from Europe and North America, but agricultural landscape and management looks quite different in different regions. And that means that we may not be able to transfer the knowledge from different regions since they look different. And this has also been recently highlighted by Adriana de Palma and her co-authors in this papers in scientific reports that shows that bees respond differently in different regions to land uses. And their results suggest that global extrapolation of models based on geographically and taxonomically restricted data may underestimate the true uncertainty, increasing the risk of ecological surprises. And this actually means that we need to do research in different regions. So, yeah. That was the wrong, no, okay. But from tropics, we know a bit, and we know that forest is often important for pollinators. It provides food resources and also nesting places and nesting material. So forest is often good in the tropics, but we know very little from sub-Saharan Africa because very few studies have been done there. So we don't really know about the relationship with forest and agriculture here, or with bees and pollination services. So I've been working in sub-Saharan Africa in Ethiopia, in the southwestern part of the country, and here there are still quite a lot of forests, actually. I've been working in this area here, and the dark green areas are forested areas, and the more light green are agricultural land, wetlands, and grazing land. From the ground it looks like this, so it's very heterogeneous. You find a mixed land uses. So here are like grazing lands, a lot of forests, smaller croplands, and there's also some wetlands back here. And the agricultural practice is traditional, small scale. Cereals are mainly grown in larger fields away from the houses, or larger, quite small, but in fields away from the houses. And vegetables and fruits, and perennial crops are mainly grown around houses, and we call this area the home garden. So the need for pollination services is mainly within the home garden areas. And there are two main seasons in this landscape. We have the rainy season, which is very green, and we have the dry season, which is more beige or overgrazed. And it's not only the general appearance that differ between these two seasons, it's also the resource distribution for bees. In the rainy season, there are most herbs flowering, and also the annual crops are flowering during the rainy season or in the end of the rainy season, whereas most trees and perennial crops like coffee and fruit trees are flowering during the dry season. And these trees and fruit trees are more patchy resources in the landscape, whereas the annual crops and the herbs are more evenly distributed over the landscape. So we wanted to include the aspect of season when we wanted to evaluate how forests cover affect the bees. The food resources are very important. So to do this, we worked in 28 home garden sites, and since we wanted to evaluate the impact of forest cover, we worked in home gardens with different amount of forest in the surrounding. So we worked in some home gardens with a lot of forest in the surrounding, and some with almost no forest, and the intermediate ones. And we surveyed the bees with pan traps, one of each color, and with vane traps. And the vane traps were hung in trees, and the pan traps were on the ground. And we sampled in both the dry season and in the rainy season. The dry season we sampled for a bit more than 2 months, and in the rainy season a bit less than 3 months. And these are the results. So, forest cover is on the x-axis. Had a positive influence on both bee species richness, here, and bee abundance. It was quite weak. It was not very strong, but it was a significant effect. And the open circles that are data from the dry season, and the filled circles are data from the rainy season. And there were no difference in the impact of forest cover in the different seasons. But the trap catches were quite different between the dry and the rainy season. So the gray ones are from pan traps, and the white bars are from the vane traps. And the catches were quite similar in the pan traps, but you can see that we had much more bees in the vane traps in the dry season than in the rainy season. And if you recall that I told you that in the dry season, it's mainly trees and perennial crops that are flowering, that may explain that more bees are foraging at tree height in the dry season, and maybe more foraging in the, or they're not foraging at tree height in the rainy season. And you should also look at this y-axis here. This is the mean number of bees collected per trap per home garden during the whole sampling time. So if you look at this one, it means that it was 1 bee collected in 1 vane trap for 3 months. It was very, very low abundance. I didn't really expect that. And we also saw it was a clear turnover in species composition between the rainy and the dry season. So this is the dry season, this is the rainy season. And we connected that to the traits, so it was more small species active in the rainy season than in the dry season, and it was a tendency to be more active big bee species in the dry season than in the rainy season. And it was also more soil nesters active in the rainy season. So we continued to do a pollination experiment to evaluate pollination services in the landscape. So we sowed rapeseed in the home gardens, and we assigned the plants to 3 treatments, controls, bagged ones where we excluded pollinators, and pollen additioning where we hand pollinated the flowers. And we followed the plants until they were ripe. We harvested them, counted the fruits, dried the seeds, and counted the seeds and weighed the seeds. So our hypothesis was that the control plants would increase, the yield from the control plants would increase with increasing forest cover, like the bees did. And the other treatments, the ones with pollen added and the ones with pollinator exclusions, they wouldn't be affected by forest cover, but The plants with pollen added would have the highest yield, and the ones with pollinators excluded would have the lowest yield. But what we found was that forest cover didn't affect the results at all. And we seem to be in this part of the spectrum. Because here are the results then, and if you look at figure D here, so that's the number of seeds per plant, we find no significant difference between the ones with pollinators excluded and the controls, showing that we didn't have any pollination services. And we got almost double the yield on the plants where we added pollen, where we hand-pollinated plants. So that shows it's a great pollen limitation in the area. And the yield parameter mainly affecting this are the fruit sets of the plants. The number of seeds per fruit capsules didn't vary that much. It was about one more seed in the fruits from the hand-pollinated ones, and the seed weights didn't differ between the different treatments. So, my main findings was that abundance and species richness of bees were positively related to the surrounding forest cover, but it was very low abundance of bees in the whole landscape. It was low pollination services and very high pollen limitation in rapeseed throughout the landscape, and this was not related to surrounding forest cover. And rapeseed is very common in this landscape. I didn't say that before, but it's grown in every second home garden, so it's a very common crop. And extra pollination almost doubled the rapeseed yields, so that will really affect the farmers. So my questions then, is there a general, future questions, is there a general pollen limitation in the landscape, or is it only for rapeseed? Does the pollination services differ between the different seasons? How can we increase bee abundance and pollination services in the landscape? And how is the status for pollination services on other altitudes in Ethiopia and in other sub-Saharan countries? And I think these are quite urgent questions, because the area grown with pollinator-dependent crops is steadily increasing. And if the area of pollinator-dependent crops are increasing in landscapes like this, with very few pollinators and low pollination services, we will have huge yield gaps, and maybe that will lead to more land being converted to agricultural land than actually is needed. So, okay, thank you for listening, and I also want to thank all people that has been involved in this project. And if you want to use the data, it's freely available here in the DRIAD Digital Repository, so you can follow that link. Okay, thank you. Yeah, we have been thinking about different explanations. They are not using pesticides. They're using some herbicide, but the pesticide use is very low, so that couldn't be the explanation. We tried to use trap nests for one of my experiments, but it didn't really work because it was a lot of aggressive ants going into this nest. I don't know. I have been starting to think about if ants could affect it, because the ants are very abundant. But yeah, I don't really know, actually.
Following on from that, do you know what do you have any idea what the natural abundance of pollinators is, and how many of the plants there are dependent on bee pollination services, or is that?
Well, that's a good question. This is a coffee growing region, so it's a lot of coffee there, and I think that's partly dependent on pollination. And but you were asking about the wild plants, or about?
Well, I'm just kind of asking about your baseline, because it looks like you've either got a already degraded landscape, maybe where you've lost pollinators, or you've got a landscape that doesn't have very many pollinators. So
it would be very nice to disentangle that, if it's because it's a heavily grazed landscape. So even if it's very heterogeneous, it's very grazed. So during the dry season, there could have been herbs, but there are not because it's just overgrazed, even though most of the herbs are flowering during the rainy season. So I think like the grazing pressure could affect. But yes, there are a lot of plants that needs pollination. So yeah.
Yeah, my question was essentially following on from that. From your photographs, it didn't really appear that there was a herbaceous community at all. Are there native brassicas in that part of the world? I'm just wondering if any bees would even want to go to brassica if it's not part of the local community.
There are some indigenous brassica species that they also grow for consumption, but that's indigenous, so it should come from there, and that's also grown in every second home garden.
What time of year were you doing the hand polishing experiments? Was it during the dry season or wet season?
It was during the wet season, or in the end of the wet season, yeah.
You mentioned that many of the local plants are in flower then. Couldn't this be that the pollinators are being drawn to other flower sources and away from the home gardens?
We surveyed the flowers, the flower abundance and the diversity, and we didn't get an effect on the treatments. So I would have expected that it would affect the controls, but not the other treatments, but we didn't find that. But we found that in general, the plants in home gardens with high floral abundance got lower yields. But it didn't affect the different treatments, so I don't think it's, yeah.
Well, I'd like to thank Ulrika, and I'd like to thank all of our speakers, and especially for keeping so well to time and providing us with such an interesting